What we're going to do today is we're going to change gears a little bit, since I'm basically been in the pharmaceutical industry serving clinical trials for a long time now. And what I wanted to do was to bring to you a white paper which is put together by a bunch of individuals across the industry to a problem that's known to serve clinical trials. And it has to do with the measurement of biomarkers and using multiplex assays. So I'm going to try to walk you through that. So the white paper was published last year. Hang on, we'll get this fixed here in a second. And if you look at the individual names there, you get representatives from the pharmaceutical industry, the CRO industry, academia. There's some folks from different government agencies there. So this is a reflection of a concern that's seen throughout the industry, okay? This paper took, here we go. This paper took about 2 years to put together. If you can imagine all the different discussions that had to take place with everybody's busy work schedule. So it's kind of a labor of love, and I'm kind of excited about bringing that to you today. So a couple of things, we broke up the talk into a couple of different sections. So one is to give you a jet tour of multiplexing, okay, just to set the stage for exactly what we mean when we say multiplexing, because different people have different ideas of what that means. We want to set that stage. I'll talk to you about some of the unique challenges that are that happen with multiplexed assays that do not occur with single analyte measurements, okay? We'll talk about some solutions or actually recommendations on how to fix that. I don't know what I'm doing with the microphone, but we're gonna be fine. And then just kind of bring everything back up. So to set the stage, we first have to narrow the scope a little bit. Okay, so we're restricting this to what we call ligand binding assays. If you've not seen that term before, it's a confusing term that I first encountered straight out of graduate school, wondering what they're talking about. You're thinking antibody-based assays, but believe it or not everything in an assay uses an antibody. So there are all kinds of ligands out there. So the industry in the pharmaceutical industry refers to these as ligand binding assays, okay? Now, what is multiplexing? So I just want to get this straight here, okay? It's multiple assays performed together with the same sample, and you have to measure them at the same time. And that's the critical piece. Some of the technologies out there will take the sample and use microfluidics and branch it into different chambers and measure the analytes separately. That's not multiplexing. Okay, that's not what we're talking about. We're talking doing everything together at the same time in the same vessel, okay? So why would you want to do that? And I'm sure all of you are familiar with these type things. We're just gonna put them out there. Well, it's a savings on volume, and resources, okay? But you have to take into account that multiplex assays are much more complex than a singleplex assay, okay? We live in a world of commercial kits. I swear I'm not doing anything funny under my jacket. So we live in a world of commercial kits. So people graduating from schools right now really don't know how to develop assays. All they know is I'm gonna buy the kit from your favorite vendor, Walmart, Target, R&D Systems, Biorad, whatever it is, and I'm just gonna run the assay, okay? So we have a wide variety of things that are available to us. There are also a wide variety of places to outsource the work to because everybody's resource constraint, okay? However, when you buy kits from different vendors, you have to understand that some of them really know what they're doing and some of them really don't know what they're doing, okay? And it's up to you to determine whether you have a reliable vendor or not. And the best way to do it is to call tech support and find out who you're talking to. And that's a good way to kind of evaluate whether you've got a good vendor or not. And I know the ones here are fantastic, so I'm not talking about you, right? Okay, so let's move on a little bit. So I've got 2 or 3 introductory slides here just to kind of set the stage. There are basically 2 flavors of multiplexed arrays. There's something that we call planar arrays, and there's something called a suspension array, which is where Luminex falls. So a planar array is basically the spotting of a capture reagent, a ligand, on different areas of a single vessel, like a well. Okay, yeah. Okay, so a planar array. So what you're doing here is you're taking your capture ligand and putting them in a different location within the plate. Okay, and I've color-coded those here. And then what you do is you perform the assay on that surface. Okay, so in this case, you're taking your planar array, you have your sample with all the different analytes in it, and what happens is those reflective analytes will bind to their respective capture reagents, okay, on that plate. You give it a wash, and most times you get rid of the excess sample. You come back with a pool of detection reagents that have some sort of tag, and you let them complete that sandwich, and then you have your array. And you know what you're measuring by the location within that well, and the signal strength that comes from that tag tells you how much you actually measured. Pretty straightforward, right? So, suspension arrays or bead-based arrays, which is what our favorite one is, which would be Luminex. What you then do now is you take some sort of particle, like a polystyrene bead, and you have some mechanism to differentiate these beads from one another. So Luminex uses different fluorochromes, okay? And on the surface of those beads, you put those same capture reagents that you do on your planar array, except you put them on the surface of beads, okay? And you take that bead mixture and you mix it with your sample, and the exact same thing happens again. These analytes are captured on the respective bead sets. Give the beads a wash. You come back with a pool of detection antibodies that carry that tag, and then you run this bead mixture through your instrumentation. The bead set tells you what you're measuring. The tag tells you how much you're measuring. So these two ways of multiplexing are a little bit different from one another, and both have pros and cons, okay? But it's just important to understand that. So when we're writing something, recommendations to the industry on this, we have to address both of these, okay? So I highlighted yellow Luminex here. There are many different other platforms and technologies that are currently out there that use a bead-based multiplexing. Okay, you have Quanterix, you have Bioscale, and of course Luminex, and there are others. So for planar array, you can be looking at something like an MSD assay, Ocean, Randox, Genelite, and they all differ from one another, usually by the planar array surface or what they're actually measuring as far as the detection tag. They all have different properties and things like that. So multiplexing covers a multitude of different technologies and ways to go about this. Okay, this is a slide that's been around for a while, but it basically shows something kind of interesting, and I'll show you how this works. So this is it's actually a 10-plex assay, but the 10th graph didn't fit too well on this, so we made it a 9-plex. So what you're looking at here are 9 different assays using the same exact antibody pairs, the same exact calibrator materials, the same samples, except they're put on multiple platforms. So here we put this on Luminex, we put it on MSD, and we put it on Searchlight, which is now the Ocean, a rare. And what you're looking at here are just standard curves, okay? And what you can see here is that even the same reagents perform a little bit differently across platforms. And you can also see that you can go back and forth between some of these platforms pretty easily, okay? And you get different binding properties and different results depending on which technology you actually use, okay? But it just opens up the discussion that there's many ways to do multiplexing, and you need to find the technology that's best for you, okay? And that's what this really shows. We won't talk about what the actual data means and who wins and who doesn't win, things like that, because it's kind of hard to see. But what I can tell you is that they all did very well. And the claims that one is superior over the other is really doesn't really hold up most of the time, okay? So that's what multiplexing is to us. That's the scope that we're going to be talking about. So let's talk about some of the things that are unique to multiplexing now, okay?
The first thing we need to talk about is something called specificity. So I'm going to be getting into a lot of terms that we use for clinical validation of assays. So some of you may be familiar with them, some of them maybe not. So specificity, what exactly is that? Well, basically, it's the ability of an assay to capture only its analyte of interest, okay? And you need to first know, does your assay do that well, okay? And an example would be maybe one analyte is captured on one of the multiplex assays, but it would not be measured by the second assay or the third assay or whatever else is being multiplexed. In other words, each individual part of that multiplex panel is specific to its own analyte, okay? Most of the time, these things are done in buffer, and we all know we live in a buffer world. So when you have clinical trial subjects coming into, they always give buffer samples, right? Everything works in a buffer world, which is not quite there. So most of these specificity experiments are done in the context of buffers and purified proteins. It doesn't necessarily mean that the assay is going to behave, okay? But we need to talk about that anyway. So there's specificity. And this is an example of a measurement of IL-23, okay? And what we did was we took the assay by itself, which is in that white bar there, and we put the assay in the context of these other cytokines, and we look for cross-reactivity. In other words, you put IL-23 by itself, none of the other assays would pick up that analyte. So the IL-23 assay was very specific for its particular analyte, which is what you want, right? So how do you measure specificity? So you go and you buy this multiplex kit. How do you determine that these assays are specific? Well, one of the things that's used is called the missing man technique or something like that. And basically what you do is you add all of the analytes except one, and you ask the question, what happens to that specific analyte? Is that one bead, that one assay, you shouldn't have any signal at all, and versus the other multiplex assays should all give their signal, and vice versa. You can just add a single analyte, and then the only thing that you should get a signal from is that one analyte assay that's specific to that, and you can check cross-reactivity that way. This is that same multiplex assay. And as you can see, it's not always clean like you think. These are binding affinities for the capture antibody here. And what you're looking at here across the top is you're taking these different cytokines and putting them in there one at a time. And you can see that some of the capture antibodies, like IL-6, will actually recognize IL-1 beta. Whoops, didn't know that. That one kind of shocked up on me because I thought my kit that I bought was very specific and I knew what I was measuring. Well, sometimes you don't know what you're measuring, okay, but you think you do, and that can skew your data, okay? So you have to do these types of experiments so that you understand what your assay can and can't do, okay? So that's specificity, and everybody understands that, and that's great. But there's something else that occurs in a multiplex assay. It's called crosstalk, okay? And basically what happens is the signal from one assay can interfere with the signal in other assays that are being multiplexed with it, okay? The biggest problem with that is probably in planar arrays, where you're looking at the emission of light from all of these different spots. And sometimes you have something called blooming, which will occur where you have the signal is so strong from one analyte, it actually carries over into the other analytes around it, okay? It's also called bleed over leaching. Sometimes the vendor does a good job of this, sometimes they don't. And it really tends to occur when you have a high expression of one analyte being multiplexed with an analyte that's at a very low signal. And that's when you really begin to see it. So all of these planar arrays, regardless of what they tell you, have this effect. You just need to know how big of a deal it is for your multiplexed assay, okay? And what you do is to really test it, you add your analytes all but one at a very low concentration, and you spike in a high concentration of one of your analytes, and you'll be able to see it. And you might be surprised what you thought you were measuring wasn't really what you were measuring all along. That's when the fun begins when you're dealing with clinical trials, right? Okay, so that's crosstalk. So here's an example that was provided by some folks at Genentech. You're looking at 4 different assays that are being multiplexed together here. And what you're seeing is they are measuring cross-reactivity of each analyte by itself, and they're expressing that in terms of a % cross-reactivity. And you can see that there is some cross-reactivity that's very small in some of these analyte pairs and not in others. So because they were looking at low levels, what they decided to do was to break this assay up into 2 2-plexes as opposed to running them all as 1 4-plex. Okay? But they did their homework on it. So, great. So you have specificity, and you have crosstalk and interference, but what are some other things that are encountered with multiplex assays? Okay. Well, one is you've got a clinical sample and you've got a 10-plex assay that you're going to run it. What dilution of your sample are you going to run? Because one dilution might be perfect for one analyte, but it will not be optimized for all of the other analytes. And you can't have it both ways, because if you end up running 10 different dilutions, then what's the point of doing multiplexing in the first place, right? So you have to come up with an optimal solution for a multiplexed assay that's different from if you ran the analyte by itself. Okay, so it's a compromise on what your optimal dilution is going to be. And this is an example of an MRD experiment, what we call the minimum required dilution of a sample. And what you're looking at here is a graph where you're taking different dilutions of your sample and you're measuring the response back. And as you can see, there is a minimal dilution that's required for this analyte in order to get a reproducible result. You can't just add straight sample to your assays all the time and get reproducible results. Okay, so this is something that we all know that we have to do, but you have to do this in a multiplex assay with all of the analytes present to be able to decide on what dilution you're going to use. And sometimes you can't find a good dilution. Sometimes you end up running multiple dilutions, or you break up the multiplex assay accordingly, okay? But it's just something that you need to go through. So here's another issue. I told you that the ability of an assay to measure something specific in buffer is great, but that's like shooting fish in a barrel. How do you know the assay can perform in its real matrix or serum or blood? Okay, it's one thing to measure interleukin-2 in PBS. It's another thing to measure it in context of serum. Okay, so that concept is called selectivity. Okay, and let me give you an example of a multiplex assay that's been out in the literature for a while now. This is an assay that measures A-beta 42, tau, and phospho-tau. And the manufacturer claims that it's very specific for A-beta 42, okay? So one of the other analytes present in CSF samples is A-beta 40. It's just a little bit smaller version of it. And this graph basically shows if you add A-beta 40, which the kit's not supposed to recognize, and you keep adding increasing concentrations, you get no signal. And if you add a fixed amount of A-beta 42, you get a reproducible signal. So the kit's specific for A-beta 42, right? Off we go, here we go. We're going into Alzheimer's trials now. This is a great biomarker. Bob's your uncle. Everybody goes home happy. What we then did was we said, well, you know, CSF, cerebral spinal fluid, has these other peptides in it, and they can be at fairly high concentrations. So what we did was we took a fixed amount of A-beta 42, and we started increasing A-beta 40 to see what happens. Well, something magical happens around 4,000 to 5,000 picograms per mil. Suddenly, your ability to measure A-beta 42 tanks. And what's going on here is the A-beta 40 is interfering with the measurement of A-beta 42 because the manufacturer didn't put enough antibodies in reagents. They didn't titer it correctly. And what was happening is people were using this kit in Alzheimer's trials to determine if their drug had efficacy or not. And they were reporting out lower levels of A-beta 42 in their samples, and it may not be a drop in A-beta 42, it may be an increase in A-beta 40. They didn't look at that because it's selectivity. So when you do a multiplexed assay, you've got to look at selectivity to make sure your assay is performing like you expect it to, okay? All right, so we're got to get you back on time here. So we're going to move a little bit quicker. Lot-to-lot variability between kits and assays is a real problem, particularly if your assay is going to be used over multiple years, like an oncology study, okay? Here is something that we had to live through. What you're looking at here are calibrators, okay, used to generate a standard curve used to measure clinical samples, okay? And each dot represents one run. Remember, you're going to draw a line between these different values to create your standard curve. I bet you can probably spot where we had lot changes. Yeah, it's pretty scary actually. Right in the middle of a phase 2 clinical trial, one of these lots had one of the calibrators was way off. And so when we were trying to draw a calibrator curve here to measure our analytes, the curve shifts. And it often occurred at these low ends instead at the top end. And it's all because the lot-to-lot variability with these calibrators was not being controlled by the manufacturer. And it creates a nightmare because lot 1 is gone. You only have lot 2 available. You're in the middle of your clinical trials. How are you going to now link the second set of samples to the first set of samples because your assay has changed? Okay, so how do you control this? Well, there's not a lot you can do, really. You can try to buy out one lot. You can try to come up with a fudge factor. But you need a statistician's help, and hopefully you can call that vendor and let them know that there's a problem with the consistency of their kits. Okay? All right. So, a couple other things that are in this white paper that I didn't talk about. We don't have a lot of time to really go through them. But it basically makes some recommendations on things that you should do. One of them is having QC samples. Okay? QC samples should be representative of your clinical trial samples. You should have them on all of your assays, and you need multiple levels of them. Okay? And that serves as a measure of consistency across your sample population. And if the assay stops working, it also tells you that the assay is starting to break down. Okay? Now, finally, we're going to add 2 more slides. I'm going to quit. What do you do if you're running a multiplex assay and one of the analytes fails? Do you throw the whole thing out? Probably not. Our recommendation is to keep the data from all the other assays that did pass, throw out the one that failed, repeat the analysis of that sample, but ignore the first set of data, the ones that pass, because you don't want duplicate results for your samples, and just focus on the one analyte that did fail the first time. If that fails again, chances are you got a problem with that particular part of the multiplex, and you need to be on that phone or talking about whoever made these assays for you. Okay. So, cross-reactivity, crosstalk, MRD, selectivity, those are things that you need to pay attention to. This is the group of individuals that's associated with AAPS, or American Association of Pharmaceutical Scientists. It came out of this focus group, and there's a lot of other things going on. This is just the beginning. This paper is not the end all. It's just the beginning of the discussion. So there's a lot of work left to be done. There's a lot of things that we need to continue talking about, and it's not all completely solved. So with that, thank you very much.